No problem. Thanks very much. Yeah, I agree. It's a very good segue from Helen's talk about what can be done when data are collected in a useful and meaningful fashion. I'm going to focus much more on diseases of aging and what can be done with those. The reason that we focus on complex chronic diseases is because they account for roughly 80% of healthcare spending, and they are the primary drivers why healthcare and in many cases social care is becoming unaffordable. When you look at the primary costs in the US health system, you'll see musculoskeletal disorders, diabetes, metabolic disease, cardiovascular, respiratory, a whole bunch of communicable or infectious diseases, mental health, neurological disorders, all massively outstripping oncology as sources of or sinks for funding. And I'll call out diabetes separately, and I've got an example of that coming up, talking about some of the things that we're doing in that space. But I want to go back a little bit and just compare and contrast the way that the world has moved forward in that oncology space versus these complex diseases. And back in about 2000, I was working with the first round of the Human Genome Project. In fact, I've been working with it for about 7 years at that stage, and there was a huge sense of optimism that understanding the genetic makeup of patients was going to be very useful in providing new solutions. And in fact, that's come to pass. What we've done is turn cancer from an anatomically defined disease where we would give patients with breast cancer the same drug just because they had tumors in their breast into one that's defined from a molecular standpoint. Sorry. Um, and here we analyze the genetic makeup of a tumor and give a therapy according to that. Unfortunately, the frustration is that that hasn't worked as well in cardiometabolic, um, in respiratory, in women's health, in neuroscience based disorders. And so that's what Precision Life set up to do. Like Helen, we take a wide range of data sources. So here, genomics, clinical history, um, transcriptomic, proteomic, epigenetic data, imaging, epidemiology of the patient, the environment that they live in, and a range of lifestyle information. These come from a very broad range of sources, and as we've seen, these all have their own formats and their own ways of handling data. What we use that for is best represented in the center here. So this is early onset Alzheimer's. Everyone whose data has ended up on this graph has exactly the same diagnosis. They show up with the same symptoms in the clinic. And yet, decades of experience says that they're not going to respond to drugs in the same way. Um, and this is what we're trying to get under the skin of. So the colors here in the clusters represent different patient subgroups with a different molecular cause to their disease. And in fact, we can see that the anti-amyloid drugs, which are very much in favor at the moment, this blue group, they represent the biggest gene target, the biggest mechanism driving disease, but it's only still relevant to about a third of patients. And there are many other ways of getting a diagnosis of early onset Alzheimer's through different mechanisms. And those patients will respond to different drugs. So understanding this is at the basis of being able to treat disease much better and indeed predict and prevent those diseases from reaching a point where they become as serious to a patient as you would expect, and therefore as expensive to treat in a healthcare setting. So As a business, we have studied about 50 different diseases, covering a broad range of therapeutic areas, with a broad range of symptomology. But these account, as I say, for about 80% of healthcare spending. I'm going to give you a case study in a disease that has currently is not really recognized as a disease by many clinicians, even though it clearly is. It has no diagnosis, it has no therapies, and we don't even know which tissues in the body are involved in the disease. Just to show you what can be done now with these types of data approaches. Um, this is a clinical, a genetic stratification of myalgic encephalomyelitis, or chronic fatigue syndrome. So this is a profoundly debilitating disease, like long COVID, but a much more severe form. And it affects, it's estimated, around 100 million patients around the world. So very high prevalence, albeit at different levels. What you're seeing, what we were able to do is to identify for the first time and replicate genetic signatures that are associated with the disease. And in fact, we have about 200 mutations here, which in one shape or form are associated with the disease. And we've clumped them together, clustered them by the patients in which they co-occur. So by itself, this is a groundbreaking result. But what we're also able to do is to correlate those clusters with the patients in which they co-occur to see whether the way those patients are presenting in the clinic is consistent with this form of disease. And here, the green group represent patients who have defects in their ability to recreate energy stores in cells from their mitochondria. And so you would expect that if that is the defect that they have, these would be the patients who would be presenting with profound fatigue, especially after exercise, this post-exertional malaise. And in fact, that's exactly what they present with. As opposed to the group on the left, the blue group, who have defects in neurotransmitter biology, they basically can't produce serotonin at the right level. And so you would expect, and in fact it turns out to be the case, that these are the patients who have cognitive impairment or brain fog, and they have mood disorders and sleep disturbance. So from a disease that's never had any understanding, we've been able to tease apart and use existing clinical evidence to validate the mechanisms driving this disease. So where can we go with this? Well, the theme is around prevention of disease and healthy aging. So what I want to talk about is where some of this biology takes us into processes that we can see are actively protective against complex chronic diseases. So taking this MECFS example, we did something incredibly naive. We went back and said, how many times can we see these genetic risk signatures in patients, which is on the left here, versus controls, which is on the right? And if there was no distinguishing power in our genetic signatures, these distributions would be exactly the same shape. But as you can see, they're quite radically different from each other. And this means that we have good predictive value or good odds ratios from the data. In fact, the predictive value is very similar to the predictive value of genetic tests like BRCA at predicting risk of breast cancer. So this is really exciting, and we can reduce this to practice into a saliva or a blood test, and I'll come back to that in the next section. But what it also allows us to do is to start to say, okay, now we have a very good risk signature that can show whether a person is at risk of this disease. Let's go and screen an entire population. And here we've used the 500,000 patients in UK Biobank. I keep saying patients, I should say people. who don't have the ME/CFS disease diagnosis, and let's evaluate what their genetic risk score is. Now, many of those will have a low disease risk. They simply don't have the susceptibilities that drive towards the disease. But some of them will have really high risk factors. They'll have had all of the exposure triggers, they'll be as old as possible, but they haven't got the disease. So in some way they seem to be resisting getting the disease, even with all of those risk factors. The question is why? So now we can subtract all of the disease processes, and we get left with a set of genes and drug targets which we believe may be actively protective. In other words, they are processes that are working in the background of your cellular biology to stop those disease triggers leading to a full-blown form of the disease. And this is clearly incredibly exciting. Pharma companies have been chasing these types of targets and have had very little success in doing that. We can now do this systematically. And it means that we can look forward to a new generation of precision targeted prophylactic drugs. And of course, as we're bringing in new therapeutic mRNA vaccines, we can anticipate having a vaccine that's delivered into, for example, the muscle tissue, which turns your muscle cells into little factories producing proteins that are protective against some of these key disease drivers. And this is super exciting. In fact, we've seen some of these in ME/CFS itself. We have, for example, found the same gene in 2 independent runs, which is involved in the transformation of tyrosine up to dopamine, which is an important neurological driver of some of the symptomology of ME/CFS. And through one of our clinical partners, which is why I'm in Salt Lake City, there's anecdotal evidence that patients receiving drugs that affect this target just coincidentally, because they're being given them for different diseases, they're actually the ones who are responding better and have less severe disease and have the potential for quicker recovery. So we are testing. We've kicked off a clinical trial of 1,000 patients with our partners, the Metrodora Institute in Salt Lake City, to test a series of these. Firstly, we're trialing the diagnostics. So for the first time, we'll be able to predict a patient's disease risk well ahead in many cases of them actually getting the disease or its symptoms. And we're evaluating the diagnostic accuracy here. But we're also bringing in a series of drug repurposing candidates that match the particular mechanisms that we can see in individual patients at high prevalence. And we're recruiting small groups of patients to trial these medicines to see whether they have a disease modifying effect. And this is incredibly exciting because it offers us a route to shortcutting the typical 12 years and $2.5 billion of putting these drugs on market. We're aiming to read out the trial this year. It only started at the beginning of this month, so 7 or 8 months of trial, and get these medicines to patients that for the first time will offer them some symptomatic relief. Now, we're aiming to do this across many of the diseases that we highlighted. I wanted to go back to type 2 diabetes. This is a $2 trillion problem around the world. And that $2 trillion, 3% of GDP in the US spent treating diabetes. It's not spent on metformin or even insulin. In fact, it's spent mainly, 80% of it is spent on treating disease complications. This is when patients go into cardiovascular events like heart attacks and strokes. They get renal failure and need dialysis or transplant. They get dementia, they get blindness, or need amputations. At the moment, there's no way of predicting who is going to have those most severe forms of disease and therefore cost the most. So we've run that study. We found 6,000 patients who tolerated diabetes for decades without undue problems, and we compared them against 3,000 patients who went on to develop serious complications very quickly after diagnosis. So we wanted to separate out that complication signal. And we have been able to develop a model that allows us to predict at the point of diagnosis which disease trajectory an individual patient is likely to go down. And this is really important because it buys us a 5-year window between initial diagnosis and the onset of these complication symptoms, into which we can deploy tailored interventions, monitoring, lifestyle changes, and even therapies to reduce the progression of some of those symptoms. These interventions are well known to work. Just looking at blood pressure on a biannual basis is enough to reduce kidney failure by 80%. The problem is they're just too expensive to deploy across everybody because about 80% of patients don't need this level of intervention. So being able to do this is a way of saving a huge amount of money. It may benefit everybody. You know, we can pick up patients who would benefit from drugs that reduce the onset of chronic kidney disease and therefore present an opportunity to reduce dialysis and transplants. The problem is this drug at the moment is only prescribed in relatively late stage renal failure. Wouldn't it be great to prescribe it earlier to preserve a patient's kidney function such that they may never experience the symptoms of kidney failure? Which brings me to my final slide, which is I was asked to talk about AI. A lot of the methods I've been describing pull heavily on AI. But you will all have heard of large language models and some of the things that they can do. So one of the projects that we're involved in with a very large health system in the US is starting to use their clinical notes as a predictor of those patients who are showing very early disease symptoms that might be associated with being at risk for one of these diseases. That gets us to a potential at-risk cohort. This is a very quick and dirty screen. These patients don't necessarily have the disease. There may be other reasons for that symptomology showing up in the clinical record. But if we now go and test those patients genetically with the type of signatures that we've been describing, we have confirmation not only that they're showing symptoms in the clinic, but also their genetics backs up that they're very likely to have these diseases. And these would be candidates for early prescription of medicines, of high monitoring, and changes in their lifestyle that could bend the curve away from that disease risk. And I think this is a trend that we're likely to see in the future. So with that, I will stop and say thank you very much again for inviting me to speak. Apologies for any background noise that may have come through here.
Thank you very much, Steve. Do we have any questions for Steve? Fascinating material. Love to work with you, and very excited about the deep dive with your data. It's something that it's all close to our hearts, but we don't know quite what to do with it.
Yeah.
Can I have a question? And it's on the diversity of the number of people. We have 6,000, 3,000. So what is the diversity? Because as we all know, NCD affects people of brown, black origin more than other people.
Yeah, it's a fabulous question, and one that we struggle with all the time in these approaches. The UK biobank population is about 93% white British. And obviously diseases affect people of different backgrounds and ancestries in very different ways, also different demographics. It's one of the reasons why getting into the US offers us an increase in diversity, and we are very actively looking for partnerships with patient populations around the world in order to increase that diversity. But technically, the methodology that we use translates much better across different diverse populations. I can offer you an example from endometriosis, where we found 10 times as much signal, explained 5 times as much disease variance, and our results replicated 9 times better than the best current gold standard analysis that's been done, simply because we recognize that complexity and the fact that different populations have different drivers of disease.
Getting a lot of
Sorry.
wind noise.
Yeah, apologies, it's very windy here.
Can I just ask? I mean, I think it's fascinating and inspiring. Often, I think we've heard a lot about interventions and prophylaxis to prevent disease, and obviously identifying susceptible populations is absolutely core. Often these lifestyle interventions require compliance and understanding and education within the patient populations. And as somebody who's very much geared to trying to help people help themselves, I was just wondering whether you have any other alliances or integrated ways of doing that, because prophylaxis requires engagement, doesn't it?
It does. No, I mean that's extremely fair. I think with things like type 2 diabetes, it's very difficult. Sorry, I'm just going to shield my mic a little bit. It is very difficult, but in a variety of the diseases that we work in, we're actually working with the patient foundations very closely in order to understand the patient experience of disease, the things that they care about, the things that are most troublesome to them. And I think one of the things that is very useful in this context is personalization of advice. You know, this is your form of the disease. These are the things you're most likely to be susceptible to. These are the things you can do to actively prevent your disease, not 5 a day, exercise more, stop smoking, stop drinking. You know, that public health message, I don't think gets through. Personalization does in a much better way.